Based on what we covered, when we compare the two synapses, you will see that the two are very different and have their respective nuances. As far as this video goes, we will not extensively go back to electrical synapses because they are not as utilized in the body as chemical synapses. And to be quite honest, in this introduction I've covered the bulk of what we know about them. So with this being said, let's get better insight on how our knowledge on chemical synapses came about by considering the synapse between the motor neuron coming out of the spinal cord and the skeletal muscle that it innervates. When we take a deeper dive into what the skeletal muscle is, you will see that the muscle is made out of many muscle fibers, and those are what the motor neuron innervates. This synapse between the muscle fibers and the motor neuron is called the neuromuscular junction. The neuromuscular junction obviously has very important impacts on the contraction of muscles, but as I said, in this video we want to understand the basis of where our understanding for chemical synapses came from, and for that reason, we won't discuss muscle contractions in this video. This topic will be covered in a video about the motor system. We will only consider the neuromuscular junction as this simple system of a motor neuron transmitting a signal to the muscle fiber to model chemical synaptic transmission. Now, one might ask, what is so compelling about the neuromuscular junction that we want to base the chemical synapse model on it? The first advantage of this synapse is its size. In comparison to synapses in the brain, which are measured on the nanometer scale, the synapses at the neuromuscular junction are on the micrometer scale, which makes them about 1,000 times larger. The second advantage is its simplicity relative to synapses in the brain. Indeed, the neuromuscular junction operates on a simple system of one neurotransmitter and one ionotropic receptor, whereas systems in the brain can become very complicated as many neurotransmitters and many different types of receptors can get involved. The third advantage of this synapse is its accessibility, and this should be obvious from the first point given that they are considerably bigger and usually more isolated. These three factors together make the neuromuscular junction a very suitable site for analysis and allow electrophysiological recordings to be made without damaging the structure. Now, the way I will go about this section will be a bit different than how it is typically covered. Instead of first going through the experiments that give insight into the process and then constructing the model, I will first go over how chemical transmission at the neuromuscular junction is currently understood, and then we will look at the experiments with a sort of introspective lens to understand how they've managed to build what we know today. So, for terminology's sake, the many branchings leaving the axon form what are called synaptic boutons, which essentially correspond to the sites of chemical transmission. The synaptic boutons are enwrapped in Schwann cells, the same type of cells that cover the axon. The sum the region covered by these boutons is usually referred to as the end plate. Now, let's consider one side of transmission, which looks a bit like this. Alright, first, let's discuss how the neurotransmitter is packaged in the vesicles by considering this small patch of the terminal. The neurotransmitter inside these vesicles is acetylcholine. This neurotransmitter is synthesized in a rather straightforward reaction. The enzyme choline acetyltransferase catalyzes the formation of acetylcholine from choline and acetyl-CoA. Acetyl-CoA is made from the metabolic oxidation of glucose by the mitochondrion, and choline enters the cell by the choline symporter that carries sodium inside as well. To get a transmitter inside the vesicles have a vesicular acetylcholine antiporter that uses the energy from proton export to get acetylcholine inside. In a later section, I will explain in more detail how the proton gradient is generated and what other important proteins the vesicle carries. Note that every vesicle has this transporter, even though it's not shown on the schematic. As you can see, everything from the synthesis to the packaging of the neurotransmitter is directly done at the terminal to ensure efficiency. With this step covered, we can now consider how the transmitter is released. The release of acetylcholine follows exactly the same steps as we've established previously when we discussed the general properties of chemical synapses. The action potential causes the opening of voltage-gated calcium channels, calcium enters, and causes fusion of vesicles. The exocytosis of acetylcholine then causes them to diffuse in a cleft and bind to receptors. The receptor on which acetylcholine binds is called the nicotinic Acetylcholine receptor. This receptor is a pentameric ionotropic channel. At the neuromuscular junction, the distribution of subunits is 2 alpha, 1 beta, 1 delta, and 1 gamma. I mentioned this distribution because the nicotinic acetylcholine receptors in the central nervous system have a different distribution, 2 alpha and 3 beta. The channel has 2 binding sites for acetylcholine that are located between the alpha subunits and the neighboring gamma and delta subunits. To open this channel, the 2 binding sites must have acetylcholine inside. When the channel opens, mainly sodium and potassium flow through according to their driving force. Thus, the nicotinic acetylcholine receptor provides a path for both potassium efflux and sodium influx. The pore size of the channel has been found to be considerably larger than the previous selective channels we have discussed, and thus, it explains why sodium and potassium can both flow. Even calcium can flow through this channel, and for that reason, it is usually referred to as a non-selective cation channel. I will later explain what experimental technique allows to discover which ions go through the channel when we have a better understanding of some prior concepts associated to it. On a side note, I want to mention that the reason this channel is named the nicotinic acetylcholine receptor is because nicotine is a substance that is capable of binding to the channel and opening it such that ions can flow. Such compounds that can bind to channels and open them are referred to as agonists. Another example of an agonist is obviously acetylcholine, but usually it isn't referred to as an agonist because it is the main substance that physiologically binds to these channels. We would technically say that nicotine is an agonist to acetylcholine for this receptor. On the other hand, you can also have compounds that bind to the channel and do not open it. Such compounds are called antagonists. 2 common antagonists for this channel are alpha-bungaratoxin and curare. These compounds are often used to reduce the action of acetylcholine receptors in certain experimental conditions, and we will later cover some experiments that use curare. Back to the schematic, now that we have a good idea on how these postsynaptic receptors function, we can now understand that the opening of these nicotinic acetylcholine receptors by the binding of acetylcholine allows sodium to enter and potassium to exit the cell. It turns out that the channel is a bit more conductive to sodium, which causes a net depolarization that passively propagates in the muscle fiber. Something I haven't mentioned yet, which is one of the most notable features of the neuromuscular junction, are the multiple folds it has on the postsynaptic side. The reasoning behind these folds is that they considerably increase the surface area in which the receptor channels can be embedded. Accordingly, the acetylcholine receptors are concentrated near the crests of these invaginations, while voltage-gated sodium channels are concentrated in the troughs. This simple organization allows the neuromuscular junction to have a low activation threshold, such that the depolarization coming from the receptors easily cause the opening of voltage-gated sodium channels and the subsequent firing of an action potential at the muscle fiber. Remember that this action potential in the muscle fiber ultimately leads to muscle contraction. But we know from our personal experience that we do not want our muscles to constantly contract, so there must be a system in place that takes care of terminating the signal and stopping the opening of the receptors. The way the signal is terminated in the neuromuscular junction is with the aid of acetylcholinesterase. Acetylcholinesterase is an enzyme anchored in the basal lamina, which is a meshwork of collagen and other extracellular matrix proteins, here represented by this blue zigzaggy line. The main catalytic function of acetylcholinesterase is to rapidly hydrolyze acetylcholine into acetate and choline. From this, choline can be directly recycled by the presynaptic terminal to make more acetylcholine, but more importantly, the neurotransmitter is degraded and can obviously no longer interact with the receptors. Now that we understand how the synapse at the neuromuscular junction functions, let's see what experiments helped pave the way for this model. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. On the right, you will see the informational resources that I've used to produce this video. Thank you again for watching, and I'll see you in our next discussion.